Imagine a security guard, right, at like a massive, sprawling museum.
Okay, I'm picturing it.
So when everything is peaceful, they walk the perimeter in a standard, recognizable uniform, just casually keeping an eye on things.
Right, standard patrol.
Exactly. But the second a threat appears, an intruder, a fire, literally anything dangerous, they instantly change into heavy, compact, tactical gear to deal with the danger.
Oh, wow, okay. Instant wardrobe change.
Yeah, and you can look at the guard and immediately know their operational state just by what they're wearing,
Because the visual cues are so obvious.
Right. And for over a century, neuroscientists believe that microglia, which are the resident immune cells of your brain and central nervous system, work the exact same way.
Oh, absolutely. It was one of the most fundamental rules we learned.
Like if a microglia was ramified, meaning it looked like this complex, highly branched oak tree, it was considered homeostatic or resting. It was the guard in the standard uniform.
The peaceful state, yeah.
But if it pulled those branches in and looked amoeboid, like a compact, blob-like sponge, well, it was activated. It was in tactical gear, fighting disease. We assumed it was entirely binary.
It is a very comforting biological assumption, too. I mean, in science, we inherently like things to be visually categorized.
We want to put things in neat little boxes.
Exactly. You look at a shape under a microscope, you categorize it, and you assume you immediately know exactly what that cell is doing based purely on its morphology. It makes the microscopic world feel predictable.
But today, we're going to shatter that assumption completely. Because in this deep dive, we are exploring a really groundbreaking 2026 paper.
It really is a paradigm shifter.
Yeah, published in Nature Aging by Huns and colleagues at Stanford University. And our mission today is to look inside the branches of these microscopic cellular trees.
We're going far beyond just the shape.
Right. We aren't just going to look at their external shape. We're going to map exactly where their genetic instructions live inside the cell, how they operate in like unprecedented detail, and uncover the slightly terrifying truth of what happens to them as the human brain ages.
It gets pretty dark when we get to the aging part.
It really does. So, okay, let's unpack this. We're talking about seeing a cell's physical shape and its internal genetic state at the exact same time. How is that even technologically possible?
Well, the methodology here is really what makes this entire paradigm shift possible, because historically, if you wanted to read a cell's genetics, you essentially had to perform a biological blender trick.
The blender trick.
Yeah, you would grind up the brain tissue.
Oh, gross.
I mean, you would get all the genetic data, sure, but you completely lost the spatial context. You lost the physical shape of the cell because, well, you destroyed it to get the data.
Right, the uniform is destroyed, so you only have the instructions left.
Exactly. But Hans and his team circumvented that by combining two highly advanced techniques. First, they used merfish.
Merfish. Sounds like a colorful ocean creature, but I'm guessing it's an acronym.
It is. It stands for Multiplexed Error-Robust Fluorescence In Situ Hybridization.
Wow. Okay, you have to break that down for me.
I know, it's a mouthful. Think of it piece by piece. Multiplex means we are looking at many things at once.
Okay.
Fluorescence means we are making specific molecules light up. And in situ literally means in its original place.
So not in the blender.
Right, no blender. So instead of grinding up the cell, they use glowing probes to light up 500 specific mRNA transcripts, the genetic instruction manuals, while they are still sitting perfectly intact inside the tissue.
That's incredible.
It gets better. Then they combine that genetic mapping with IBA1 immunostaining.
And what does that do?
This is a targeted dye that perfectly outlines the physical 3D shape of the cell across multiple microscopic layers.
Oh, I see.
Yeah, by layering these two techniques, they managed to analyze 3,970 individual microglia.
Almost 4,000 cells.
Across 7 distinct brain regions. And they compared 3-month-old or young mice with 24-month-old or aged mice.
So we finally have the technology to read both the mind of the cell, its genetics, and the body of the cell, its physical shape simultaneously.
That's the perfect way to phrase it, the mind and the body.
Which immediately begs the question, right, what happens when the mind and the body completely disagree?
Well, to answer that, we have to look at how the researchers organized this massive mountain of data. When you have thousands of cells and hundreds of genes, human eyes can't just spot the patterns.
No, you'd just be staring at glowing static.
Right. So they relied on an algorithm called UMA. That's Uniform Manifold Approximation and Projection.
I've heard of UMA used in data science, but How does it work in this specific biological context?
Imagine taking insanely complex, high-dimensional data, like hundreds of different gene expression levels, and squashing it down into a flat 2D map.
Oh, almost like taking a massive spreadsheet of demographics for a whole country and turning it into a map where similar people are grouped into the same zip codes so you can actually see the neighborhoods.
That is a perfect analogy. And they created two completely distinct UMAP maps for the exact same set of cells.
Two different maps for the same cells.
Yes. The first map grouped the cells strictly by their transcriptomics, their genetic state, and they found three main zip codes, or states.
What were they?
Homeostatic, transitioning, and DAM, which stands for disease-associated microglia.
Okay, so DAM is the disease state.
Correct. Then the second map grouped those exact same cells by their physical morphology, completely ignoring the genetics.
Just looking at the uniform.
Right. And here they found 5 distinct physical shapes on a continuum. They labeled them C1, which is the most amoeboid, compact, blob-like shape.
The tactical gear.
Exactly. All the way up to C5, which is the most ramified, highly branched, tree-like shape.
And this is where the massive aha moment of this paper happens, doesn't it? Because when they cross those two maps, comparing the genetic zip code with the physical shape's zip code, the 100-year-old dogma just completely fell apart.
It fractured. The UML overlap revealed a large subpopulation of dam-like microglia.
The disease ones.
Yes. Now, genetically, these cells were screaming that they were in a full-blown disease-fighting state. They were highly expressing classic disease-associated genes like ITGAX, AXL, and SPP1.
But wait, if they're expressing disease genes, the old rule says they should look like a compact C1 blobs, right?
That is what everyone assumed for a century. But almost 30% of these highly reactive disease-associated cells were highly ramified.
30%.
Yeah, they were in the C4 and C5 shape categories. They looked exactly like the peaceful, resting tree-like cells, even though their internal genetics were fully activated and responding to pathology.
The uniform no longer dictated the behavior.
Exactly.
I really have to push back on this, though, because how does that functionally make sense?
What do you mean?
Well, let's say a cell is actively fighting a disease, like clearing out amyloid plaques in a brain with Alzheimer's. Why wouldn't it pull its branches in, become a compact little blob, and just focus on eating the debris?
That's the logical assumption.
Right. Why would it expend the immense energy required to keep this vast, sprawling branch network open while simultaneously maintaining an inflammatory, disease-fighting state?
The functional logic here actually points to the incredible sophistication of the brain's immune response. The working theory is that these cells are performing dual roles.
Dual role?
Yeah. They might need that massive sprawling branch network to surveil a huge area of tissue, staying in contact with dozens of neurons at once while still being biologically primed to release inflammatory signals or perform targeted cleanup at the very extreme tips of those branches.
So the amoeboid blob shape is great for localized, heavy-duty eating, what we call phagocytosis.
Yes.
But this highly branched, disease-associated cell is acting more like a wide-net coordinator.
Precisely. It is coordinating a broader response across a larger physical space during a disease state.
That is absolutely wild. Okay, let's just pause and synthesize this to make sure we are all tracking. I'm going to do a quick mini recap here.
Go for it.
Bullet 1, microglia are the brain's immune cells. Bullet 2, for a century, the unbreakable rule was a branchy shape equals a resting cell, and a blob shape equals an active cell. Bullet 3, this new data proves that up to 30% of disease-associated highly active microglia are actually highly branched. It completely flips the script.
It really is a fundamental rewrite of the textbooks.
So for you listening, think about your own field of work or study. When has a deeply held, visually obvious structural assumption completely blinded people to how something actually functions under the hood? It makes you wonder what else we're getting wrong just by trusting our eyes.
The visual bias in science was incredibly strong. But, once we accept that physical shape alone doesn't dictate a cell's function, we have to look deeper.
Right, we have to find out what actually does control it.
Exactly. We have to ask what does dictate it. And to find out, we can't just look at what genes the cell is expressing. We have to look at the geography of the genetics.
The geography?
Yeah, we have to map where those exact mRNA transcripts are physically parked inside the microscopic space of the cell.
The geography of genetics. I love this concept. I think of this as the cellular postal service.
The postal service. Okay, I like that.
Yeah, imagine the cell isn't just a balloon filled with liquid, but a massive, highly organized organization. The soma, which is the central cell body containing the nucleus, is the main headquarters and sorting facility.
Right.
And the processes, which are those long branches reaching out, are the delivery routes. Now, if the recipe, the mRNA for a specific crucial action, is just sitting in the central sorting facility, it's totally useless.
It's just sitting on a desk.
Exactly. It needs to be packaged and shipped out via the delivery routes, so the final product, the protein, can be built exactly where the action happens, out at the furthest edges of the tissue.
This concept of spatial compartmentalization is absolutely vital to understanding the cell. And the Stanford researchers took their MERFISH data and algorithmically separated the cell into two distinct compartments, soma and process.
Headquarters and delivery routes.
Right. And they discovered that the mRNA is not just floating around randomly. They identified 14 distinct soma-enriched genes. And these were primarily linked to the creation of endocytic vesicles.
Okay, let's define it really quickly for the listeners. Endocytic vesicles, what are we talking about practically?
Basically, they are the cell's internal sorting, recycling, and digestion bins. When the cell swallows up surrounding molecules or debris, it traps them in these vesicles inside the main body to process them.
Oh, so it makes total sense that the instructions for those bins stay in the central headquarters.
Exactly.
Yeah.
But out in the branches, the delivery routes, they found 57 process-enriched genes, and these were heavily linked to mitochondria.
The powerhouses of the cell.
Yes, the localized power generators. The branch tips are so far from the central cell body that they need their own local energy supply to function.
That makes sense.
The process genes were also heavily linked to the structural scaffolding needed to maintain those complex branches. So the instructions for energy and structure are physically shipped out to the edges where they are needed most.
And this delivery system isn't just a neat little organizational quirk, right? It is so rigid and reliable that the researchers were actually able to predict the physical shape of the cell purely based on the mail that was out on the delivery route.
They did. They utilized a machine learning algorithm called a random forest classifier.
Random forest.
Yes. By looking only at the mRNA floating out in the processes, completely ignoring the genetics inside the main cell body, the algorithm could accurately predict the overall physical shape of the cell. It achieved an AURC score of 0.76.
Okay, AUROC area under the receiver operating characteristic. For those of us who aren't data scientists, how good is a 0.76?
Well, if you build a model that just randomly flips a coin to guess an outcome, your AUROC score is 0.5.
Okay, total chance.
Right. And 1.0 means your model has perfect, godlike clairvoyance. So, in the highly noisy, chaotic world of biological data, hitting a 0.76 means your predictive model is incredibly strong.
Wow.
It proves that the localized instructions physically sitting in the branches are what dictate the shape of those branches.
And there are a couple of superstar genes driving this entire prediction model that are just fascinating. Let's introduce them. SLC1A2 and PINK1.
Yes, very important genes.
We need a way to remember this. Let's make a mnemonic for you listening. Think of SLC1A2 as the super long cell gene.
Super long cell, I like that.
If the Cellular Postal Service successfully ships the mRNA for SLC1A2, the super long cell gene, out to the branches, that cell is going to be super long, highly branched, and ramified.
What is truly brilliant here is the biological paradox of that SLC1A2 gene. It codes for a protein called GLT1, which is a glutamate transporter. Now, canonically, in older literature that relied on grinding up the tissue, elevated SLC1A2 was known strictly as an activated disease marker.
Oh, really?
Yeah. But here, with intact spatial mapping, we see that having its mRNA shipped out to the branches correlates heavily with a highly ramified, supposedly resting morphology.
So simply having the disease gene doesn't make the cell a blob.
No.
Sending the disease gene to the branches allows the branches to perform active, disease-mitigating work while staying fully spread out. The delivery system determines the function.
You hit the nail on the head.
Let's do another quick recap. Bullet 1. The mere presence of an mRNA transcript isn't enough. Its physical location matters just as much.
Okay.
Bullet 2. The cell's central hub handles the internal recycling, while the branches get the instructions for local power and structure. Bullet 3. Genes like SLC1A2, our super long cell gene, act as local instruction manuals out in the branches to dictate that complex shape.
Exactly.
It really makes you think about systems design.
Right.
If you were designing a complex organization or system, which core instruction manuals would you hoard at central headquarters, and which would you distribute to your furthest outposts?
It's all about efficiency. The cellular system is built on incredible efficiency. But the most alarming part of this study happens when we subject this beautiful, highly organized cellular postal service to the ultimate biological stress test.
Aging.
Aging.
Yeah, this is where the story gets a bit dark. Everything we just discussed about this pristine organization, that was observed in young 3-month-old mice. But the researchers also mapped the brains of 24-month-old mice.
Elderly mice.
Right, and we see complete structural decay of the postal service.
The numbers are stark. I mean, in the young brain, we saw robust compartmentalization, 16 soma-enriched genes and 50 process-enriched genes. The instructions were flowing smoothly to the outposts.
Like a well-oiled machine.
But in the aged brain, the delivery system collapses. Aged microglia dropped to just 8 soma-enriched genes.
Down from 16.
Right, and only 23 process-enriched genes.
Down from 50. Wow.
The necessary instructions for power and structure simply are not making it out to the delivery drivers anymore.
And you mentioned earlier that it's not just a few lost packages. The entire network of workers seems to fall apart. How do we actually measure that microscopic collapse, like mathematically?
The researchers utilized a spatial algorithm known as Ripley's K and H functions.
Okay, that sounds intimidating. How does it work?
Think of it like drawing microscopic circles. The algorithm takes a single mRNA transcript and draws a tiny circle around it, essentially asking, how many functionally related transcripts are sitting right here in my immediate neighborhood?
Okay, like setting a radius.
Exactly. In young cells, the answer is many. The transcripts are tightly co-localized. They cluster together like highly efficient assembly lines, producing proteins right next to each other.
But in the aged cells?
The co-localization drops massively. The transcripts are physically isolated. They drift apart.
Like workers in a sprawling factory who used to stand shoulder to shoulder on an assembly line, seamlessly passing parts down the line.
Yes.
But now, because of aging, they are scattered in entirely different rooms, trying to shout complex instructions at each other through the walls.
And obviously, the system grinds to a halt.
I have to imagine that this massive internal breakdown completely changes the actual mission of the cell, right? What is the mechanism there? Why does it change?
If we connect this to the bigger picture, the shift is dramatic and reactive. In a young, healthy cell, the central soma networks are proactive. They focus on healthy metabolism and cytokine production.
Cytokines being the chemical warning flares the immune system uses to signal for help.
Exactly. And out in the branches of a young cell, the focus is on precise protein localization and synapse pruning.
Meaning the branches are carefully sculpting and refining the connections between neurons to keep the brain fast and efficient.
Right. But in old age, because that localized energy and structure fail, the soma abandons production. It shifts toward chemotaxis, which is essentially desperate movement and migration.
Desperate movement. That sounds awful.
And the branches, they go into a full panic. Without their localized instructions, their genetic networks shift heavily toward endocytosis and catabolic regulation.
Catabolic meaning breaking things down for raw material.
Tearing things apart, yes.
Let me make sure I'm visualizing this mechanism correctly. The young cell is an elegant sculptor. It is building, surveilling, and signaling.
Beautifully put.
But the aged cell, because its internal supply chain is dead, lacks the energy to maintain the wide net.
Yeah.
So it abandons the careful surveillance. It goes into a desperate scavenging survival mode. It stops being a precise sculptor and just starts tearing down its surrounding environment to survive.
The lack of local energy and structure forces a shift from proactive maintenance to reactive, destructive survival. The cellular infrastructure collapses. Yet, what is truly remarkable is the resilience hidden in this decay.
Gosh.
The random forest classifier, that predictive model we talked about, it actually still works in aged cells.
Wait, really? Even with the decay?
Yes. The accuracy drops slightly
from
a 0.76 to a 0.72, but it still functions.
Which means that even while the assembly lines are drifting apart and the cell is panicking, core structural genes like SLC1A2 are still fighting tooth and nail to define the cell's architecture. They're still trying to do their job in a failing system.
It's an incredible biological effort to maintain stability.
Let's do one final synthesis of this breakdown. Bullet 1. Aging physically destroys the cell's ability to transport mRNA from the central hub out to the branches. Bullet 2. Because of that, the genetic assembly lines physically drift apart. Bullet 3. This forces the cell's entire purpose to shift from proactive surveillance in youth to a reactive, scavenging survival state in old age. A perfect summary. It's a profound parallel, really. Does the breakdown of spatial organization within a single microscopic cell remind you of the aging of human infrastructure? like a sprawling city whose transit system is slowly, inevitably failing, forcing the distinct neighborhoods to just fend for themselves.
The infrastructure of the cell absolutely dictates its capacity to function, just like a city.
This has been an unbelievable journey today. We started by completely overturning a 100-year-old biological dogma, proving that a cell's physical shape doesn't simply equal its disease state.
Right, the uniform doesn't dictate the mission.
That security guard can wear the standard uniform, have their branches spread wide, and still be in full combat mode.
Exactly.
And then we zoomed in further to physically map mRNA within specific branches, discovering the highly organized cellular postal service. And finally, we witnessed the tragic but fascinating mechanism of how time dismantles that intricate transit system, turning a precise cellular sculptor into a desperate scavenger in the aging brain.
And exploring this mechanism raises a final, vital question for the future of medicine. Well, currently, when the medical field thinks about treating neurodegeneration or brain aging like Alzheimer's disease, the focus is heavily on pharmacology that turns specific genes on or off. We try to increase or decrease expression.
Right, editing the instructions.
But this paper shows us that the disease state is heavily driven by a spatial failure, a failure of mRNA trafficking within single immune cells.
Because if the recipe never leaves the central sorting facility, it doesn't matter how many millions of copies of the recipe you print. The branches still starve.
Exactly. So what if turning genes on isn't enough? What if the next massive frontier of neuromedicine isn't about altering the genetic code itself, but simply inventing therapies that fix the cell's internal postal service?
Oh, wow.
Helping the aged cell deliver the mRNA it already has to the correct branches so it can rebuild its assembly lines.
Fixing the transit system to save the city. Not changing the instructions, just fixing the roads. That is a massive paradigm shift to leave you with. Thank you for joining us on this deep dive into the microscopic, bustling universe of your own brain. Take a moment today to appreciate the billions of tiny, highly branched security guards working overtime, constantly shipping packages back and forth in your head. Keep wondering, keep questioning assumptions, and we will catch you on the next deep dive.